Okay, then let's start. Can everybody hear me? Can you just do a final sound?
Yes.
Okay.
Yes, I think it's all good.
Very clean.
Okay, good. Single molecule spectroscopy, maybe just a word on that. Well, it just means you're working on single biomolecules, but spectroscopy can mean many things. And today we're going to focus on force spectroscopy. So I did a lot of my experiments in my PhD pulling on single proteins, and I'm continuing work along those lines here too. Before we get into the slides, I thought we will start off with a video so that you have some sort of imagery in the back of your minds that you can then refer back to as we go through some technical details. So for spectroscopy, one way to do this is by using optical tweezers. So you have beads trapped into laser beams, and in between these beads, you have strung up something that you want to look at or that you want to pull on, basically. This little white blob, can everybody see the mouse?
Yeah.
Yes, good. This little white blob is a protein, and it's strung up to two linker molecules that then are attached to the beads. Now, this protein has some dimension. It is sitting, the whole system is sitting in a solution at room temperature, which is why you can see it wobbling about. It's basically to simulate the Brownian motion. And you can now see also from the familiar double helix, you might recognize this as DNA linkers because we can't attach the protein. For these kind of experiments, we can't attach the protein straight away to the beads. Now, but to make it a bit simpler, we take all the faff away and just look at the cartoon of the protein and have the DNA pretty much as strings. To exert force on this complex, we just start moving away one bead from the other and basically stretches. But the protein itself has some resistance to stretching. So it will start pulling both beads out of the traps until, of course, there is a certain point where it can't withstand the force anymore, and then it unfolds, which means that the force relaxes and the beads move back towards the trap centers, towards the laser centers. Now, if the force is not too high, the protein can also spontaneously refold. And then again, the beads are moved out of the laser foci, they move back as it relaxes, and so on and so on. Now, at the bottom of the video, you can now see a trace, which is basically as you sit at a constant trap distance, and as the protein unfolds and refolds, you can see high force signals for when the protein is folded, and low force signals for when the protein is unfolded. And you can pretty much do this forever, but of course we don't do this forever. So let's swap to the presentation. Oops, sorry, not on the first slide. There we go. Now, for those of you who haven't really heard about or dealt with biomolecules in a long time, I'll just recap some very basic features of both like biomolecules in general and also folding of such biomolecules. Then we'll discuss a few basics on the force spectroscopy side of things that are necessary to understand the data that I'll talk about in the second half of the presentation. So I'll first show you a few examples from the literature and also from my own projects that I did while I was at Cambridge to show you simply how the data can look like. And then I focus on the protein that we currently have some trouble analyzing because of heterogeneity or messy force data. And of course, to be able to deal with this stuff, we also need to deal with artifacts that arise due to the instrument. And we have to discuss those as well, because those have to be removed. Let's start with the building blocks of life, pretty much. So you might remember the sugars, amino acids, and fatty acids. And of course, I mean, here in this picture, we also include the nucleotide, which is a combination of different organic and inorganic molecules. Now, the fatty acids form the membranes of cells and also the membranes of cellular organelles, such as the mitochondria or the nucleus. The sugars you can find often on cell surfaces, but they also, as I said, form the basis of the nucleotides. So the DNA, which encodes our genome, and RNA, which floats around the cytoplasm. And then you have the amino acids. And these form, these are the basis of a variety of proteins that, for example, stabilize the cell through the cytoskeleton, perform lots of functions within the cell that are crucial to survival. Now, the cell, of course, doesn't just produce these on its own, it puts them into 1D polymers. So in the case of nucleotides, we have the desoxynucleotides, which are synthesized into DNA polymers, and we have the ribonucleotides, which are synthesized into RNA nucleotides. And as you might remember, DNA can be transcribed into RNA. These kind of RNAs can then further be translated into proteins, which are then composed of a chain of amino acids, but they can also remain on its own and perform functions on their own. Now, these, for a lot of systems or for a lot of functions in the cell, not all of them, but a lot, these molecules have to adopt some sort of 3D structure. You are probably familiar with the DNA double helix, but maybe not so familiar with the kind of more complex conformations RNA can attain. Proteins come in a humongous number of shapes, forms, and sizes, but here I've shown two, one that contains mainly sheets, what we call sheets, or sheet-like arrangements, and then a protein that contains mostly helices. And for the remainder of my talk, I'm mostly discussing proteins that are purely helical, or I also have an example of RNA. Now, we have a lot of data with a lot of advances on the bioinformatics side, or no, on the sequencing side of these molecules. So we have a lot of data on the 1D arrangement of these building blocks. And also we have now a increasing amount of data, both experimental and computational, of these 3D structures. And in the next slide, I would discuss more the computational approach, because I thought, that might be a bit more interesting to you. So of course you can predict these, or ever since the first structures came out, people try to predict structures, like how can a certain amino acid sequence, for example, in case of proteins, give rise to a specific 3D structure. Up until recently, I would say the Baker labs and the Zhang labs were dominating really the field. And I've used both of their servers at least. And these two, labs base their software more on knowledge-based approaches. So homology modeling, where you match the protein that you're interested in to a known target, and then assume that the structure will be somewhat similar, or through threading, where you take your amino acid sequence and thread it through structures to see whether it's physically possible for that sequence to obtain that structure. And then, of course, you have the most ideal case where you can predict the structure based on first principles, but due to computational constraints, this is only really possible for very small proteins, so less than 100 amino acids. With the hierarchical approaches, I haven't really dealt a lot myself, but basically they're going more in steps. They're first looking at how the amino acid sequence falls into small motifs and then into higher order structures. But then, of course, something new came along and AlphaFold basically overtook them all, simply, I guess, because of how it works, the deep learning, machine learning approach. It now totally outperforms everything, and a lot of us are playing catch up now to see, and are starting to play around with it. Now, on the one hand, we have these vast amounts of sequencing data, and we are starting to, with both experiments and computational approaches, gain more information about the 3D structures proteins can obtain. But what happens in between? So how do we get from the sequence information to the 3D structure? And that is what folding is all about. Now, why do I call it a folding problem? Well, simply because people in the field call it a folding problem, because originally in the 60s, they thought, well, we'll solve protein folding pretty soon. Unfortunately, that was not the case. So 70 years later, we still hadn't really solved the problem. We've gotten along closer, though, have solved the folding problems for individual cases. And yeah, and we have found out that there is certain driving forces. For example, you have electrostatic interactions, hydrophobic interactions, van der Waals interactions, and hydrogen bonds that stabilize structures. Torsion angles of the polypeptide, right, limit the kind of conformations it can take, because, I mean, yeah, polymers can't fold back on itself. And then, of course, you have entropy that drives everything more towards an extended state and destabilizes structures. Now, for us, it is often easiest, and for a lot of other people, it is easiest to think about folding in a sense of a more like landscape image. So if you think about a 100 amino acid protein, it would sample and think about that it could sample every possible conformation that that polymer could take, but only one is the actual functional conformation. Then you would, it would take longer than the age of the universe to sample all of these conformations, because the probability of finding this hole in the middle is really, really small. Now, but if you then start distorting this energy landscape to something that, you know, as you make contact, form something, you then go lower in energy, and then that limits the conformational space that you can sample, and then iteratively basically reach the energy minimum in your landscape. Now, real proteins, or a lot of real proteins, more look like this kind of landscape, you know, with barriers in the middle, with troughs, sometimes also traps in there. And usually there is a path, you know, that basically gets it from the unfolded state to the native or folded state in a very quick time. And this allows, this kind of energy landscape allows polymers to fold on a millisecond and microsecond timescale. Now, why do we study protein folding? Why is it important? Well, you can imagine that the environment that a protein or any biomolecule sits in, or that any kind of chemical modifications that can occur in the cell, or mutations, like we all know about mutations these days, right? So they can change not only the final structure, so the native state, but they could change the folding pathway. Now, if you imagine that we have somewhere in this landscape, maybe behind this barrier here, we have a trap. And in a normal, healthy context, is maybe 10% of the molecules end up in this trap, which means only 90% of the protein synthesized by the cell actually reach the end conformation and can perform their function. Now, if you now change the folding in a way, or if a mutation changes the folding pathway in a way that 50% end up in this trap, then of course you have a lot less molecules in the end that reach the end conformation that you want. And of course, this might then cause a certain disease. Now, at this point, then I would like to progress to how do we actually study this? Well, if you imagine, we just look at the folded state and the unfolded state. And maybe as an image, it is good to remember that, or I mean, it might be to imagine that we have something like a raw egg, which is, you know, uncooked, and a cooked egg, in which all the proteins are what we call denatured. However, this image is slightly wrong, because as you all know, you can't go back to the raw egg. Once it's cooked, which is why I'm going to substitute this with images of like chains of beads, such as this, because folding and unfolding in many cases is reversible. And a lot of our theory is based on this assumption that the folded and unfolded conformation sit at equilibrium and that it is reversible. Now, then the only thing what we do in protein folding studies is by distorting the equilibrium and pushing it more towards the folded or the unfolded state. And this you can do by changing the pH of the solution, by adding chemicals, by heating it up, similar to the egg, by increasing the pressure, or by using force, of course, to pull on it. And this is what we're going to focus on now. At this point, I would like to quickly stop and ask, are there any conceptual questions? No? Good. Then to the next point, the force spectroscopy basics. As you've already seen in the video, we have glass spheres trapped in beads. We have the protein strung up in the middle, and we have some sort of linker molecules between the protein and the beads. Now, it is important to remember that you can't just stick the protein to the beads. You have to specifically attach it. That means you have to modify your bead surface to be attached to the DNA. You have to modify your DNA and your protein for these two to react specifically, and you have to do this on both ends, of course. So just remember that we're not just, whenever I show these glass spheres, it's not just glass spheres, it's glass spheres which are covered in protein and hence are slightly bigger, slightly stickier, and that kind of limits the practicality of things. Then to explain or go through the data in more detail, I need to define some parameters. So when I talk about distance, I mean the distance between the traps, not the distance between the beads. Bead displacement is literally what it says. It is how far the bead center has been displaced from the trap, and I usually denote that with X. The extension is a property of the molecule, and it just basically says how far it is extended, like the end-to-end distance. Now, when you talk about polymers, you also have to consider the contour length. The contour length is literally just the physical end-to-end distance, or the distance, end-to-end distance of the polymer when at its maximal extension. So you can't stretch it any further than that. And in that, we have to distinguish the contour length between the DNA and the contour length of the protein, because they're both two different polymers. Now, a lot of the data that I will show you is from this constant velocity mode. So in this mode, you simply keep moving one bead away from the other at constant speed. And of course, you can then go back towards the other bead. So it's reversible in that sense. Now let's go through the steps. At point A, the construct is under no kind of tension. So we have about 0 force, but not 0 distance. Then we start moving one bead away. We start increasing the force as the tension builds up on the DNA molecule until a certain point where the protein can't hold on any longer. And then it unfolds and you see a partial relaxation of the force. Then you start pulling on this combined polymer until that is fully stretched. And this is what you see in the last part of the curve here. Now, you can pull on this molecule up to a certain point. It depends how stiff your traps are. And it depends how stiff your traps are. And at some point, you will just end up pulling the bead out of the trap, or the DNA will melt at about 60 piconewtons. You'll notice these lines here that I've drawn in, these gray lines, and they are basically fits to the data, which are based on polymer models. In this particular case, the worm-like chain polymer model that describe the force-based or force-induced extension of our polymers. So this first contour here describes the extension of the DNA, and the second contour describes the extension of the DNA protein. Let's call it copolymer. That's why, because we here in this contour, we still have the DNA response in there. That's why we often don't refer to the protein contour length directly. We talk about contour length changes because we can't probe it directly. It's always relative to the DNA molecule. Now, sometimes in the talk, you will see force plotted versus distance, and sometimes you'll see force plotted versus extension. There's various reasons for that described in the publications, but for you just to take away now is there is not much difference in terms of displaying the features that you see. Next, I would just quickly cover again the constant distance mode, just to remind you from the video. Basically, if you sit at a constant distance here, for example, here where you can equally sample the folded and unfolded states of the protein, you can then just sit there and over time see the protein in the folded state and in the unfolded state and see it fluctuating in between. Before I go on, I would also like to point out that this whole system, like a trap connected by a complex polymer in the middle, is complex also in terms of its energy. So on the one hand, we have the internal energy, which usually we consider only the energy of what we're interested in. So of the protein, think of it more as chemical energy. It's independent of force. And then we have a term that is dependent on the bead displacement or dependent on the force itself. So, however, this term is again a combination of the energy stored in the construct. So with the construct, I mean the total polymer here and the energy stored in the trap. Because of course, there's, as you displace the bead from the trap center, there's always a restoring force back into the trap center. This kind of, complexity, especially this term here, makes it nonlinear, and down the line makes it difficult to apply certain models to our data. Now, the examples that I'm going to cover are, in those examples, or these examples, I divide up into what I would call a homogeneous force response, or where you see only variation on the kind of statistical variation scale, and heterogeneous force responses, which are highly heterogeneous. So where variation is not just arising due to, random variation or random noise. And within these two classifications, let's call them, I give you some examples of folding mechanisms that are simple or very simple to see in the data, and some that are more complex. At this point, are there any questions? If I don't hear anything, I'll go on. Good, let's start with the first example, the homogeneous and simple case. Here you have a so-called leucine zipper, which are basically just two helices wrapped around each other or coiled around each other. And as you apply force, you will start unzipping this coil until it is just two coils on its own, or rather two peptides on its own. Now the force extension plot you can see here in black, the unfolding curve, and in blue, the refolding curve. In the zoom in, you can see that there is first, as it unfolds, there is like a slightly gradual transition from the native or folded state to the first intermediate. So think of this intermediate as being this coil partially unzipped to a second intermediate. Sometimes you can fluctuate in between there, but then at some point the force is so high that it goes fully unfolded and doesn't return back to this intermediate. Then as it refolds, so following the curve in blue, we do not reach the same forces, but it does refold under force. So then you, you just see you see some sort of energy loss because you have because these two folding curves are not perfectly matching, but it does refold, and then you start relaxing the DNA again. The important thing to point out here is that the variation that you see, of course, you see some variation, especially in these fluctuations between these two intermediate states, but the variation is all of a similar scale. And you can see that the hysteresis is all of a similar scale. So we have a relatively slow unfolder here because we can make out states. We can see force peaks. We have two intermediate states. And yeah, we have a relatively homogeneous, reproducible force response. Next, we'll discuss one of my projects. So these are consensus tetra-tricopeptide repeats, or let's call them short CTPRs. These are just repeats of helices. So these have exactly the same amino acid sequence. Each helix turn helix motif has exactly the same amino acid sequence, which is then in this particular case repeated 20 times. And as you stack these motifs against each other, they form this kind of superhelix. Now, the force response of these is rather unique. Here again, you stretch the DNA, and then as the protein starts to feel the force, it starts unfolding, but without ever giving us any decent force peaks, which means, or which basically is a signature of that the protein unfolds so fast that it is beyond the time resolution. of our instrument. So basically, these fluctuations that we see is not just random noise, it is a signal of this unfolding, of the unfolding of this pattern, but we didn't really know until we fit some models how exactly it, what is lying below that noise. To be able to fit any kind of models to this data, we had to severely average or bin it, because otherwise it would just take too long to run the algorithms, the fitting algorithms. And as we averaged, we also noticed that these undulations in the plateau, or these oscillations in the plateau, are reproducible between different molecules. Prior to that, we actually thought they're just some random variation, but actually these oscillations arise from the superhelix, so this whole structure turning in the trap as you're unfolding it. On top here, you can see the noise or the standard deviation of this force signal. So as we pull on the DNA construct, we stretch it. As there is a force on the beads, we constrain their movement within the trap, which of course also undergoes Brownian motion, which means that as we constrain the motion of these beads, we start reducing the noise until, of course, the protein starts doing its stuff, and then the noise goes very large up again. And at the end, when the whole polypeptide is or the whole structure is unfolded, then you start reducing the noise again. And looking at this kind of noise was very crucial to fitting our models, to doing several cross checks, even if we didn't use it for the final fitting. But it was useful to figure out what kind of filtering artifacts we had, and a few other things. So to fit any kind of models to it, just to summarize, we had to average a lot, we had to account for the tertiary structure, and the models that we used are so-called Ising models. Now, those of you who have some, who studied some physics modules in the past might be familiar with these from ferromagnetism, in which basically you have, you describe a 1D line of spins. The spin can have one energy if it's up, can have another energy if it's down, and then there is an interaction potential between these two spins that will force them to align them. Now, in the protein context, we are talking about a helix being unfolded or folded, and when it is folded, it will induce its next nearest neighbors to fold, because it is more stable that way, because it's stabilized by the interaction between the helices. Now, however, if you look at this particular protein, we have 20 individual helices in there. So we have 2 to the 40 possible states, which of course were a bit impossible for us to describe. So we had to do lots of simplifications to be able to figure out the folding pathway of these molecules. And in the end, we found out that they just unzip from the end, starting from one end, then starting from the other end. And then they just unzip until there is some sort of core left, which is this final blip here, and before they are fully unfolded. Now, next to the heterogeneous examples, first, viral RNA. Here you can see the force versus extension plot of multiple curves overlaid. You can see that there's some force peaks here, some hidden down in here, some there, and some there. So you can see that there is variation in terms of, the contour length change in a certain unfolding transition. There is variation in the force, even if we have the same contour length change. And and it's kind of, and the force peaks can be pretty much everywhere. Here you can see 3 consecutive curves, like just offset. So that show you that even within the same molecule, you can have different force peaks, different contour length changes, which kind of complicates the data analysis, because one of our aims is always, before we do anything to the system, is to establish some sort of a footprint of a particular molecule. In the refolding curves, you can't see force peaks very well, but you can still see some sort of heterogeneity, again, also in the curves that are offset. Now, these colored lines are the fitted worm-like chain models for different models or at least let's say they first fitted worm-like chain models to each of these contours, obtained a contour length, and then obtained a contour length distribution. And then they took the sequence, the primary sequence of this RNA, and modeled the RNA structure using a computer software or server called mFold, and which is particularly which is designed for RNA folding. And then they literally just looked at the structures that got spat out by this algorithm, and and then just matched the expected contour length changes from a given structure to the contour length changes that they observed in their experiments. This was only possible, of course, because they could average their data, because like in the end, like this data is not as noisy as some of the traces I've shown you before. The second example I would like to show you is Calmodulin. This is, again, a helical protein with 4 distinct domains. Here you can see the force extension traces in dark blue, the unfolding curve in light blue, the refolding curve. And from these 3 curves, you can already appreciate that the contour length changes are different and that the force peaks occur at different forces. Now, the author of this paper recently told me that originally they actually tried clustering the data based on the force extension curves, but they didn't get very far and rather did constant distance measurements, because in constant distance measurements, they were actually able to identify 6 different states. So here is the wild type or the natural protein. And here you can see the different states colored in purple. This is the fully folded state. Red is the fully unfolded state. And then there's a few states in between. Now, to be able to assign which states belongs to which of these substructures, they simply removed domain 1 or domain 4 or domain 1 and 4, and then just looked at which states remained. The easiest example is probably this one. The only thing that remains is the fully unfolded state, so the red state, and the orange state, which is visited very rarely. They could also see that certain transitions would only occur from one state to another, but then would not proceed to another. So for example, from the red state to the orange state, or you would get transitions from the red to the orange and back, but you would never see the orange progressing anywhere to any other state. And by analyzing lots of this data, they were able to draw this kind of energy landscape with a native state at the bottom. They observed 2 different folding pathways. So it could fold from either end, either first domains 1 and 2 or 3 and 4, or it could also be trapped in some traps within the energy landscape. For example, here it could only come out of by unfolding, and here it could come out of by going to this intermediate state. So to get rid of this complexity, these authors here looked at constant distance data, they measured multiple protein variants, and I would also like to point out here, they used a very high precision instrument, which allowed them to distinguish also the contour lengths of, for example, these states that are very close together in contour length. Now, this brings me to the protein I will discuss in the rest of my talk, which is PR65. Again, it is a purely helical protein, and here we have 15 repeats of helices, of helix pairs, let's rather call them like that, that are arranged not in a superhelix, but more in a horseshoe-like shape. At the bottom here, I have given you some sort of schematic of these repeats, colored in domains that we expected this protein to unfold in. So these domains, at least the rough boundaries of these domains, we have established in other experiments, not single molecule experiments. And so we were kind of expecting that we would see 5 different force peaks. But of course, we didn't. First, again, here you have the DNA stretching. Then as soon as the protein feels force, there are some fast fluctuations between 2 different states until it can't refold into this original state anymore. Then it starts in the second transition until it ruptures. Then it goes into a third transition, and then in the fourth transition that gradually goes into the, into the contour of the unfolded combined construct. The refolding then overlaps a bit in parts with the unfolding curves, for example, in this part of the force extension trace or in this part, but sometimes it also does not overlap. So this would have been easy, maybe to map out if it always was like that, but of course it was not. If you look now at several force extension traces of the same molecule, you can see that the protein never unfolded in the same way. Sometimes this third transition here is longer, sometimes it's shorter, sometimes it's almost not present at all, like for example here. And the same, sometimes in the first transition you have less noise, sometimes you have more noise. Same with the refolding. With the refolding traces, sometimes this transition towards the end here is very long, sometimes it's shorter because this other third state gets visited. Sometimes you see this spike in the front and sometimes you don't. But if you overlay these curves, then you can see that the protein, even if it is highly heterogeneous, it still goes through the same states. So these are always the same. It just differs when they are entered and when the states are exited. And similar, but not as well, they overlap for the refolding curves. Now, one problem we have is at the moment is that with such heterogeneous data, one of the first things that we kind of need to prove is that the variation that we see within a single molecule is the same or similar as the variation that we see between molecules. And that's one control that I'm currently unable to do, but that still needs to be done. Now, our questions to this kind of data were, well, of course, can we map the unfolding pattern onto the structure? How do the unfolding and refolding differ? Because partially they must go through the same bits of, or like to the same pathway, because sometimes they overlap, right? Then also one question is, can we distinguish mutants from the wild type? Because we were actually originally interested in whether certain mutations that occur in diseases change the mechanics of the structure, which at the moment we can't do because of the heterogeneity. And here I would like to point out that the mutants can change both the general unfolding patterns, so the force peaks or the contour length gains between force peaks, but they could also change the noise, so the speed at which certain domains unfold and refold. One of the first things that I did is similar to the RNA example, to fit contour lengths for the DNA only here at the beginning, and then to every conceivable point within the unfolding curve. And then of course I got averages and standard deviations for each of these transitions, or for the combined transition of that force peak. And of course we have expected contour length data from the unfolding of these domains that we've assigned it before, or combination of these domains. Now you might notice very quickly that these numbers don't really match, apart from maybe, given the large standard deviations, A2, C1 and C2 could possibly be these last two repeats within the array. But I already know from some other experiments that these two repeats definitely unfold and refold in this last bit of the force curve. So they can't be it either. I also tried constant distance measurement to be able to, see different states. But one problem that we have is that these unfolding events occur at a very, over a very narrow force range, which means that we get a very noisy data, many transitions that can occur, and you can't really assign states anymore. So my next question to the data was, of course, well, can we cluster it, right? Because if we can at least pool similar traces, in different clusters and then take a cluster average, well, can we then get something out of these contour lengths, because we won't have these large errors on there anymore, or standard deviations anymore? Because, I mean, these deviations are, well, definitely not nanometer or sub-nanometer precision that the instrument should enable us, or should give us, right? Well, as it is, it's not possible, because, well, we have a few things to consider for clustering as well. So, of course, first of all, you have the normalization. Of course, on one hand, the normalization that you are familiar with, but also in another sense, maybe more biological sense, you have to normalize to get rid of the DNA response, because in the end, you only want to look at features from your protein. You're not interested in the DNA. Fortunately, there's a way to do this. You can just remove the DNA force data, basically, through transformation, and then you could come to a plot such as this, where you now have the force versus the contour length of the protein plotted, and you have no information on the DNA anymore. Then, of course, we want to preserve the X and Y scales. Well, the Y scales, because we're interested in the height of the force peaks, and the X scale, because we need to know the contour length changes in between those, or maybe that's easier to see here. We need to know the distance between these two transitions. Then, of course, you can already imagine that, well, dealing with such, you know, with these noise from where things unfold could really affect your clustering, right? Or like how efficient your clustering is. So one needs to think about how to smooth or downsample the data. And well, of course, that then gets rid of part of what we want. So we want to see these kind of noisy transitions, because in mutations, the amount of noise in these transitions might be different. Then, so what I could imagine, for example, is that you could first, you know, totally average these curves, cluster based on these average, and then cluster the noise separately. But then, of course, there might also be things that eye by eye could not yet see in the data. So there might be things hiding somewhere that, yeah, that I don't know about, but that might still be useful for clustering, or that might pop out of an initial clustering approach. This brings me to the variation that we see and that we don't want. So artifacts that we see during the measurement or due to the instrument itself. So let's first talk about the easy things, the offset and drift. When I talk about offsets, I usually mean offsets between data sets, so between different molecules. You can have them both in the force and the distance dimensions. In the distance dimension, they're mostly due to the uncertainty in zero. So basically, zero we define as the point where the two beads touch. The problem is, well, the other way around. So basically, as the two beads touch, and if you keep moving the right bead towards the left bead, the right bead will exert a force on the left bead, and you will see the force signal of this bead increasing until this is pushed out, and then it will, it will decrease. So it gives us this kind of spike, which we then can align with what we think is zero. Now, if you remember from the basics section, from the force spectroscopy basic section, these beads are not just hard spheres, they're covered in a squishy kind of surface of proteins and all kinds of other molecules that we need to make them as the least sticky as possible, or as, yeah, the least sticky as possible, something like that. So sometimes we see a nice feature, a spike like this, but sometimes we also don't. So then we don't really have anything to align zero with. And then, of course, you have the always annoying drift. And that is usually we see within one molecule during a measurement, and then we often have to interrupt our measurement, or we can move our system away from whatever is causing the drift within the measurement chamber. But here is one example shown again of the TPRs, where we have a bit of drift, which doesn't really change the force response on its own, and we could correct for it. and we would like to correct for it, because at the moment I have to basically set all my offsets towards or correct for offsets towards one set of data and then throw away the rest. I could, of course, just pick up the light blue curve and align it by eye with the blue curve, with the dark blue curve. But well, that is not really ideal, because then it's down to, you know, the person doing that, and not really objective. So what we are still lacking is an objective way to align the data to remove these offsets and drift artifacts. Another thing that can introduce artifacts is the trap itself. So what I've not mentioned up until now is that we have to calibrate the trap, because we do not actually get a false readout from our signal. We can only measure how much the bead is deflected from the center of the trap. So we do a calibration measurement, which relies on the bead size, the temperature and the viscosity of the medium that the bead sits in. And we basically just measure the Brownian motion of the noise, fit a certain equation to it, and obtain our trap stiffness. Now, this is, of course, based on finite measurement time, which basically introduces certain filtering artifacts, etc. And basically, if any of these is estimated wrong, you can get miscalibration. And on top of that, you can also have crosstalk between the traps, which might be due to alignment, laser power, or whatever. So on the left here, you can see an example where there is no miscalibration. On the right, you can see an example with miscalibration. Now, if you just look at these force distance curves here, or the force extension curves in here, gray is always the measured or the simulated measured data. The dashed line is always the truth, so the actual parameters that were put in. And you can see that here, without miscalibration, of course, perfectly overlays. Here it slightly deviates. But what I would like to point out, a curve like this, with this kind of small error, can still be fitted by a worm-like chain model. So like experimenters can totally miss that their trap is not perfectly calibrated. But what you can do is you can, of course, measure the bead deflection noise, and you can, because of the calibration, the force signal has a calibration in it. You can calculate from the force signal what you would expect the noise to be if the force was true. Now, in a perfectly calibrated system, the calculated and the measured noise was, of course, perfectly in an overlay. In a miscalibrated system, these would not overlay. Like the rate curve here is slightly on top of the gray dots. Now, then a collaborator of ours has approached or has developed a method, a fitting algorithm that basically you just fit this measured noise, and then you can obtain correction parameters for your calibration. Then you can reverse basically any artifacts that you can see in your force distance or force extension data. On top of that, you can also have trap artifacts due to nonlinearity. So of course, our traps are not ideal. So a realistic trap is here, or trap potential is drawn here in the solid line. The dashed line is Hookean potential. Now, since we have these deviations at higher bead deflections, we also get into a range where the forces are not really linear anymore. So that means they start falling off as you get to larger extensions. And also depending on how things are calibrated, what power you're using, these can show more or less of a spread. Now, with this noise-based correction, you can correct for that as well. The correction is not perfect, but it is well enough, or at least much better than what you can see here. And basically here, this is an example of my data. You can see two sets of curves here that deviate significantly. And these were taken before the instrument was realigned and after the instrument was realigned. So that just simple instrument realignment or working on the optical table can change things, obviously. but after correction, we can get rid of most of that variation. Now, basically a possible workflow that I can imagine is, of course, well, at first drift correction, because if we can correct for drift, I can include more data. At the moment, if I have drift, I can often only use like 5 to 10% of the data. We need to remove miscalibration and non-linearity effects. And then any leftover variation that we can detect in the DNA response, we can remove by contour length transformation. And then it's basically, well, just, in quotes, just aligning the data, testing the downsampling, and clustering it. Now, basically, the things that we cannot do at the moment is the drift correction, the alignment, and yeah, the clustering might be straightforward in the end after all the artifacts are removed, but I don't know that yet, because at the moment, the only thing that I can do or have done right now is remove the DNA response, because the correction for miscalibration is relatively recent. And with that, I'm also already at the end and would like to thank my former supervisor, Laura Itzaki, who gave me the opportunity to work on these proteins, and people in Munich, Matthias Rief and his group, where I did all of my measurements, and then Johannes Stiegler, who now has his own group, and who I collaborate with still very frequently. And with that, I'm happy to take any questions.
Okay, can you hear us now?
Yeah.
Yeah, all good. Okay, any questions? I think there was a question on the chat, which was, why are refolding curves not the same as the folding curves? And does it result in a different folding structure?
Yes and no. So for example, in some systems, especially in those that fold very fast, the refolding curve is exactly the same. Like, for example, if I go, wait, let me remove the chat window. If I go to here, there. So here, these the unfolding and the refolding curves are offset because they are on average pretty much like exactly the same. In the case of, like, for example, the zipper here, or the or PR65, if I get to it. No, there, sorry. Well, they must come back to the same structure, because we can then see the same states again in the unfolding curve. So the only thing that I can say is that they probably refold via a different pathway, not that they come back to a different structure at the end. This is what I think is the most likely. Of course, I can't exclude it, but yeah, I don't think so, because I know from some other artifacts that I see with this particular protein system, like when misfolding occurs, so when different structures are attained at the end, then it looks very different.
Any other questions?
I think we could scroll out and get questions.
Okay, the only last thing I want to mention is that Marie actually has this data, and well, some of it anyway. And if anyone was interested in a project along these lines, then you can get in touch with her. I think the information is already on the course website, and you can ask any follow-up questions and whatnot. But other than that, thank you very much for the talk again.
Okay.
One small infrastructure thing. Thank you all. Most of you have now answered the